Okay, so I'm making this biology lab video for your Enzyme Lab report. Okay, this will be due by the end of the month. I guess I'll give you time. Do it as soon as you can. You want to get all your lab points in. You'll be turning it in under assignments. So I've made in the content section, there's an Enzyme Lab report section now, and it has data as well as the enzyme graph. So you should open both of those up, which I've already done. Okay, so this is it. One of them is for Excel. Hopefully you have that on your computer. And this one's for, it's a Word file that contains the data. So normally for this lab, what you would do is you would generate this data right in the lab itself. You'd use potato enzyme extract, which contains an enzyme activity. And then you would measure over a period of time, 30 seconds, 60 seconds, 90 seconds, 120, up to 3 minutes. You would use a spectrophotometer, and that would give you a number, 0.2, 0.5, 0.7. As this number goes up, that means you're accumulating product. The enzyme is working. So you can see in this last column, 0.2, 0.3, 0.4, it doesn't go up that much. So the numbers didn't go up because there wasn't as much product being made by the enzyme, so there was less enzyme activity. So we've got at 22 degrees centigrade, we conducted the assay and got this data at these different time points. Then at 40 degrees, you would have done the same thing except at different temperature, right? Increase the temperature and got generated data. So it actually, the numbers went up a little bit more at 40 degrees, which is a little bit above body temperature. Body temperature is 37. And then at 80 degrees centigrade, which is pretty hot. It's hot to touch. It would burn you if you touched it. It only went up to 0.6. So to see that on a graph, I want you to input this data into Excel, right? So I've already put in, right, 0.2, change this 0.3 to 0.5, then 0.7, 1, 1.2, 1.4, 1.5. As you put these numbers in, the graph will change, okay? Then what I want you to do is put it in for the 40 and do it for the 80. And then you'll have a nice little graph, right? The blue line will say at 22 degrees what happened, at 40 degrees what happened, at 80 degrees what happened. And you can compare the three temperatures and see, well, you know, you won't have to make any conclusions, but normally you would conclude what temperature did it work best at. And in this case, right, at 40 degrees, it worked a little bit better than at 22 degrees. The higher this number, the better the enzyme worked, right? Because the spectrophotometer is an instrument that actually just measures color. But the product produced like a brown, yellow color. And you'd have seen that in lab, had we done the lab in the lab. But this is, I think, a nice little, you know, it's worth like 20 points. So it's an easy 20 points. You put in all the data, save this file, and then submit this Excel. If you want, you could copy this and paste it into another Word file and just name that whatever. You could actually put it back into this one if you wanted to. You know, just copy the graph and put it and save this and then upload that to the assignments. If you just want to, you know, submit the Excel file, that's fine. Either way, I've never looked at an Excel file in the assignment folder. I don't think it'd matter. If there's any big issues with that, you could always email it to me. So under the assignments for the lab, this is a lab assignment, right? Just put your file that contains the graph, the finished graph at all those temperatures. You should do each temperature. You should put in the 40 and the 80 in the graph. Save it and then upload it to that folder. Okay, so that's all you got to do for the enzyme report. Okay. And I think that's it, like for the lab, right, the next thing you need to complete, be sure you do the lab practical, complete that, it's online. It'll be just like practical 1, I told you that there wasn't going to be any genetic problems that I normally have on practical 2, right, it'll be mostly, right, just like it was in the practical 1, you'll have images from the PowerPoint for practical 2, and then you have to answer questions about each of the mitosis, or about the photosynthesis lab, or the cell respiration lab. There might be some general questions, from monohybrid or dihybrid crosses that are in the lab practical. Practical 2 PowerPoint. Okay, so the information you need to know for practical 2, just like with practical 1, will come from the practical 2 PowerPoint, and there may be some quiz questions, I can't remember, that are worth 1 point, I think. I haven't, let me see. Practical 2, we've already got it set up. So 40, yeah, then there's 20 quiz questions, so they'll be worth 1 point apiece. Right, there'll be 40 questions worth 2 points apiece. That will contain, they'll be like the last time, you'll have to enter in 1 or 2 words to answer the question. Right, and I'll have to regrade it after you complete it, to check to see, it might mark it wrong. It automatically grades it, but it might mark it wrong if it's not spelled correctly, and there might be more than 1 correct answer. So you got to keep that in mind too. So don't forget the practical 2.